So I hope this recording is started.
Yes, it has been started.
Welcome everybody to our meeting. So today we will discuss the introduction to the biological part of the Kaggle competition on open problems in the single cell analysis. So today's speaker is Larisa Okorokova, and she will give a talk for us. And thank you very much, Larisa, that you agreed to present your results. So Larisa participated in the competition, which have been last year, organized by the same company. And it's a very close competition. So she is quite familiar with this topic. So thank you very much again, and please start.
Thank you very much, guys. I'm really glad to be here today. And a special thanks I want to say to Alexander, because he is supporting all this activity in the Sberlog community. So how can I make it? Sorry, I forgot how to make it on a full screen. Yeah, it's here. Okay, as I am a biologist, I actually have graduated from the biological faculty. I will give you a bit of introduction to the biological story behind this competition. So I'm Larisa, and I'm a PhD student at Université Côte at Nice, France. And if you want me to share my experience about this, I will be also very glad. Okay, so at first, I want to tell you why specifically the bone marrow cells were taken for this particular competition. The thing is that we really know for a long time, we as a biologist, we know that a lot of cells in our body which have almost complete, almost the same genome in all the tissue, in all the tissues and in all the cells. The cells might be very, very divergent in terms of phenotype. They can have very different morphology. They can have very different function. And the bone marrow cells and the bone marrow as a tissue is a very good example of this, because this particular organ have an extremely divergent cells. They can give birth to very different populations like immune cells, ordinary erythrocytes, and all this stuff. So that's why this is the first rationale to take this tissue to this competition. Moreover, we already know quite a lot about these cell populations. So there is a special thank you for some technical things like flow cytometry and other stuff which help us to research, to make a big research on this thing. So there is a whole established protocols for study on blood cells, and also we can that was actually the first single cell technology that we apply for this particular cell. So that's why we know quite a lot about them, and that's why we want to proceed with this. The next thing is very close to the medicine field, is that these cells are widely used for transplantation. And this is almost the only and the single example of the organ which can be taken from the person. And it makes almost nothing for him. So myself, I was a donor of hematopoietic stem cells. And this procedure it's a little bit uncomfortable, but I would compare this level of uncomfortable just with having a flu. So I just have to go to sometimes hospital, but yeah, nothing in comparison with other transplantation types. And also we can. The fourth factor that I want to highlight is that the samples can be taken from different people, and it's relatively safe. As I said, if you're a donor or you just do this for the research, it can be done relatively safe. Okay, so I also wanted to highlight how the bone marrow transplantation occur, because that can help us to understand why particular time course data were were taken into this competition. So we called allogenic bone marrow transplant. That means that we can take cells from healthy donor. There are several ways to do it, but we will consider the part where we put the needle into the bone and then we just push it, we just pull the bone marrow inside, we just pull away this bone marrow cells. After that we should actually clean cells a little bit, and we want to take only the cells, the hematopoietic stem cells. They possess the CD44 marker. This is very important for the medical procedure. So that is how we distinguish the cells from the cells that we don't need. Okay, we took away unnecessary cells, and then we can put them into the recipient. And the recipient in this case should be, it's almost always the case of hematology disease. So they have some bad cells which in their own bone marrow, which produce like cancer cells, and they took away all the resources from the body. So the recipient should be prepared for this procedure. Almost all the time it is chemotherapy or radiation therapy, which were before the transplantation. We ordinarily put the cells into the vein, and then the cells just go through with the bloodstream around the body. Someday they just meet the bone marrow, and then they just stay in this bone marrow and they make the new colonies. And basically the recipient after this procedure is the chimera, because the cells from the body and the cells which of the blood, they are completely different. They are basically from the different persons. So as I said, the recipient should be prepared for this procedure, and that might really mess up all the things, because we need to prepare the donor, we have to prepare the recipient. What if we just take the cells from the donor and then we store the cells, or maybe not even store, maybe we can make more cells from this, because this is hematopoietic stem cells, so they can actually divide. What if we take just a little amount, but really matters when we were saying about taking hematopoietic stem cells? What if we just take a little amount and then, or in a petri dish, we will just make them divide? That would be very nice, because we will be able to make multiple copies of this particular donor, and then we can actually use it in multiple transplantations. Sometimes recipients may have to encounter several transplantations. So I think that was the main rationality to make the time course data for this particular competition. I have not read this. It's just my, I suppose that was like this. So the cells were taken from the donor, and then they were plated in a petri dish. There was some questions in the competition chat how the cells can actually live outside the body, and biologists are quite good in this. So we actually can take the cells from the person, and we can support them alive quite a long time, sometimes even for decades. It's in case of the cancer cells. In case of the stem cells, the procedures should be very, very accurate. It's quite difficult, but we actually can make the cells alive for a really long time. And sometimes we actually try to differentiate the cells. So we want to get particular population of the cells. So maybe someday we can actually raise the whole new heart, or the new whole blood, or the new whole kidney or liver. But that will not be soon, definitely. And that's why we need the help of the machine learning, because we cannot imagine doing all this research by ourselves without the special technologies. So that is quite big rationale behind this competition. So yes, we just put the cells in a petri dish, and then we put them in a special incubator in a special media, and the cells can happily divide there and be quite quite in their place. So in different at different time points, we took the organizers of the competition took the some samples and make the single cell experiments. I would a little bit further, I will tell you what is this particular type of data that they recovered.
Larisa, may I ask you a question? So what do you think, did they took this several days from the patient, or they took just one time from the patient and then they give these cells to be, they were alive and they just put the cells to the sequenator machine on day 2, on day 3, day 4?
As far as I understand, based on this description, they took the cells from the donor only once. These procedures might be very, very painful, so I'm not sure that doctors will allow someone to make several functions in several days. I think then they put them into the incubator. So in this petri dish, they were alive and they actively divide, as far as I understand from the description of this competition, till the day 4, the day 10, sorry, and they took the samples on the different days. So that I suppose that will that this one will influence this circumstance will influence our result, because the culture conditions are not perfect, so we cannot actually suppose that the cells will be just the same cells in all the days. We try to make it, but we don't. We are not definitely sure. So yeah, I think that they just divide all the time inside this petri dish, and the samples were taken on the different time points.
So probably they put these cells at day 4 into the sequenator, then then in day 3 and day 4. So
yes.
Okay.
Part of the cells to the site-seq experiment, part of the cells to the multi-ohm experiment, and another one just to support the population in a petri dish.
Okay, maybe you'll discuss it later, but let me ask, not to forget. So do you have some expectations, the batch effect from these different days for these machines? Do you expect it to be quite big or not? I mean, batch effect, that means that if you made the
I will tell about the batch effect more precise and at the end of this presentation. Just not to spoil everything, but yeah, I expect a big batch effect just because I expect a little biological variance between these days, and I expect a way bigger effect of a batch effect just because of the technical issues. Okay, so what is next? Next, I want to describe the different data that the others provide us. So here, I want to highlight, we need to understand what is site-seq data and what is multi-ohm data. I know that yesterday, Ksenia a little bit talked about this different amount of modalities, but I want to introduce you more biological thing behind it. Why do we need to take this particular modalities? The spoiler, we just knew how to do it, but this is the best we can do. So that's why we want to understand the biological variance behind it. So I'm pretty sure that you are all familiar with the central dogma of molecular biology.
Larisa, excuse me, there is one question. Let me unmute.
Yeah, thank you. Actually, super simple question, but it's better to understand it now than later. On previous slide, the donors want to, when we have like Marrow
It seems Alexander. Sasha, you have connection problem. I didn't hear you. Can you repeat, please?
Yes, yes, it says connection is unstable. Do you hear me now?
Yes, yes.
Okay, so the question is donor 1-3 at the left upper corner and all the similar scripts. It means that actually the cells are pulled from these donors.
I need to read the description more precisely, because from a biological point of view, I would expect that this is nonsense to pull all these cells together in one dish.
Yeah, because I would think the same, but the thing is that we have.
Sasha, again, the connection problem. We don't hear you well.
I just switch off the video, maybe it will be better now. So the thing is that we have donor 1-3 on left upper, and we have donor 1-4 on down right. So probably it means pulling of the cells from different donors, and it could be actually like it's strange. Or I do not know what could it mean other than pulling, but I hope it's not pulling.
No, as far as I understand, it's just that donor 1-3 went to the train test, and another part of the donors goes to the public test. No.
So technically they are not pulled.
Yeah.
I see. Yes, thank you. So it's not pulling, it's just train test. Sorry. Okay, thank you.
There is another question from Aleja. I think there were 4 Petri dishes evolving in parallel, right?
Yeah, that would be, in biological sense of view, it would be very good decision. And I think there will be even more Petri dishes. So, you know, in the first day, we make 1, 2, 3, 4, 5, around, let's say 6 Petri dishes with maybe with doubles. And then on days, we just took away one and make the RNA extraction and all this thing. Yeah, I think that donors were not mixed up inside the Petri dish. That would be very it would be very dangerous, of course, in particular, because some cells can actually attack another one. So we can assume that some big immune fight may occur inside one Petri dish, between different donors. So no, they definitely cannot be in the same Petri dish.
Thank you very much. I guess we can continue.
Okay, thank you. So now we will go to quite, I hope, familiar stuff. This is the central dogma of molecular biology. And I want just a little bit of a reminder of this, because later I need to put this these terms like DNA, RNA, and protein into my explanation. So I we know that DNA can store in our inherited information inside every cell what we can meet. This DNA is almost the same in every cell, and the differences are due to not very good mechanisms, not because not on purpose. So it's just the mistakes that each cell can make. So that's why the DNA can be a little bit different between different cells. But basically, this is almost the same. From the DNA, the cells make a transcription process, and during the transcription process, the mRNA is released. Then this mRNA, we talk about human cells. So this mRNA will go to the cytoplasm, and with the help of the ribosome, they will make proteins. So the thing is that DNA store all the hereditary information, and proteins make all the job. So then we try to understand the very fundamental biological problem of the connection between the DNA and proteins, or more broadly, connection between genotype and phenotype. We actually need to build these bridges between DNA, mRNA, and proteins. And that's why we measure all these modalities. Okay, what we try to measure when we want to understand something about the DNA. We want to understand which particular DNA parts are accessible for transcription. So DNA DNA is very, very long molecule, and we want this molecule to be a little bit organized. If we want to have a very big genome like humans have, we want it to be organized. And what we do, we actually put this chromatin onto the nucleosomes. These nucleosomes consist of special proteins, which are called histones, and that help us to organize everything. But if we actually can put some epigenetic marks on the histones on the DNA itself. So that's why some genes can work and some genes cannot work in these particular conditions. But we are interested only on a state of chromatin right now. So what does it mean? It means that if the chromatin is actually what we call open, the transcription can actually occur. And if the chromatin is quite condensed, we call it closed chromatin. And in this case, the transcription cannot occur. I remind you that transcription is the process of building the new mRNA based on DNA complexes. So then we look in if we have this kind of data, what chromatin regions are open and what chromatin regions are closed, we hope that we can predict the expression level of the genes. And but the dependency is not very straightforward. So we actually need a lot of factors to understand, and that's why we produce so much data and ask for help from machine learners to understand, to actually predict the transcription from the chromatin state. So how this kind of experiment occur? We have a special assay which is called Rnaseq Data, and this is basically the DNA info. What does it mean? Then we have the open and closed chromatin. We can extract it from the cells, and we can put a special enzyme, I would say, which is called TN5. It's actually a transposon thing. What does it mean? If this chromatin is open, this transposon can actually cut this thing. We can extract this DNA. And in this case, you can see here the open chromatin region. It proceeds here. After the fragmentation, we still can see this region. And then when we put our reads on the genome, we can see that there is a special peak. It's actually called a peak of the open chromatin. If the chromatin is closed, transposase cannot go into this chromatin, cannot fragment it, cannot make a fragmentation, and that's why we cannot take this DNA to read in our sequenators. So that is basically the Rnaseq data what you will experience in this competition. And as far as I understand, this data is presented in beans. So in a big table, we can see the regions of the genome. They are marked with the chromosomes and their coordinates. I will talk about this particular system a little bit later. So for each cell, we know the state of this particular region. Is it open or is it closed? Like 1 or 2, 1 or 0 Boolean variable. Okay. Do you have any questions regarding the Rnaseq data?
Yeah, colleagues, I think this is something critical for understanding. So if you have some questions or you have doubts that you understand it correctly, please ask questions here. Maybe we need to give some time to. So I see the questions. Are regions precise?
Sorry, precise. What precise regions precise?
Just a moment. I'll try to unmute Andreas.
No, I have already seen this this question, but I cannot assess this, unfortunately. So I can.
So the question is really the regions have a magnitude, right? The magnitude, like the peaks here, but also the base, start and end. So I have no idea about biology, but I may be surprised that they can pinpoint exactly which base it starts and it ends. So the precise aspect is related to the start base and end base.
Well, let me give a comment. Maybe I'm not correct, but I think the position, the start and the end, are precisely defined in this competition data. When you see the names of the columns, they are something like GL or something, and then there are two big numbers. And I think that these two big numbers are exactly start and the end.
Yeah,
yes, I am not completely sure. We should clarify it maybe.
Yeah, Sasha, you're absolutely right regarding this particular data. But if I understand right, Andreas is trying to address the question, is it how good it's connected with the cell state? Am I right? So can we actually? Okay.
It's okay. It's okay. Let's move on. Okay.
I'm sorry.
Okay, but still, I think this is important for everybody, maybe to have some understanding. So, anybody who has any question, please kind of here. So, let me maybe. So there is another question. Just a moment. I'm also interested in understanding a little bit better about this TF-IDF thing. Okay,
but
then
that is interesting because I'm sorry, I'm not familiar with this term. Can you please give me a little bit more about information about this term?
This is something standard in the machine learning. So actually, it's not to biologists question; it's a question to machine learners. This is typically used in the natural language processing. It's something. IDF is inverse, F is frequency.
Inverse document frequency?
Inverse document frequency, yes.
I'm sorry, I cannot comment.
Okay, maybe we can discuss this IDF a little bit later. It's a kind of machine learning. So it's completely not biological. It's just something where it's technical thing, how they code it. So here is the biological picture. So as far as I understand, so we make something like there is special enzyme. So this is some substance which we put into the let's say into the Petri plate.
We at first we extract the nucleus from the cells, then we put enzyme, and then this enzyme makes the fragmentation.
So this enzyme destroys everything which was open from chromatin. Yeah.
No, it cannot enter the closed chromatin. So if we see the signal, that means that the chromatin is open.
Okay, so okay. Thank you very much.
Yeah, I think that I also need to highlight that the genes are quite long in our genome, and the beans, they were quite small in this data. So you might be interested in understanding the precise coordinates of the gene and some region before this gene, like the promoter region. This is the region the transcription started. So that might give you a clue, because if we know exactly the position and we know the exact position of every gene in our genome, we know the promoter region. So this is the ground truth behind this experiment. It is not always fully detected, but you can compare the coordinates of the features that you can see with the particular gene. I will tell about this a little bit more, then I will give you a little bit the introduction of the nomenclature and all this thing. Also,
sorry, Larisa, another question in the chat. So for the first peak on the plot, is it for promoter?
Almost, if we have an active gene, we will have a very broad peak that will cover the promoter and the body of the gene. You mean in this particular picture? Yeah, but that might be the promoter also. Yeah, I would say like this. But for the different genes, it would be the different signal. Sometimes you can see very broad peak, and sometimes you have a narrow peaks.
So it can be promoter, but it can be enhancer, yes, or it can be something else, yes. It's not very clear what we get. There is some signals, yes.
Yeah, that is a big biological question behind this, and that might be enhancer. That might be a very special regulatory element behind it. So we basically most of the time we use ataxic data to predict regulatory elements in the genome. And let me put this in this way. Sometimes we have a lot of mutations in non-coding regions, which are actually very tightly connected with some diseases. And this kind of experiments help us to understand that this region harbor, for example, this mutation, which can be very deleterious for the cell function. Yeah. Okay, and also I want to answer the question of Denisa Stabenko. This is it's called this base number is taken from a line reference genome and may not correspond to exact base distance in a subject genome, right? Yes, I totally agree with you. Our reference genome is a genome in a bubble, I would say, and that is exactly the truth that this particular genome can have some kind of variability from the reference genome, and that might be the case. I'm not sure that it can particularly influence the data in this competition, but yeah, from the technical and biological point of view, this is a very important consideration. And also, Ksenia asks about the nucleosome signal in ataxic data. Yeah, this is what we're talking about. So basically, we can understand where the nucleosome, where we can understand the nucleosome position here. Okay, seems that we can go further. I'm really sorry, but my Zoom cannot work properly, so I need to share my screen again. Sorry for this inconvenience. Yeah, do you see my screen again?
Yes.
Super. So next thing that I want to discuss with you is Rnaseq data. This one, I would say the most, the easiest one. So as I said, our DNA is stored in the nucleus. Then we have a transcription process, some kind of processing. I will not go into this in more detail. And then mRNA is exported into the cytoplasm and goes to the protein. With the help of a ribosome, it makes a protein. So then we want to do the Rnaseq data, which is also will be referred as gene expression data or GEX in this competition. We actually took this kind of molecules, almost all the same. We try to take only the mature one from the cytoplasm. We isolate it. And basically we send it to the sequencing, and then we can actually make the mapping between the reference genome and this in this particular read. Sometimes reads comes not from the genes. That's okay. That's because we don't know exactly what is all the functions of all the nucleotides in our genome. But most of the time we are interested in the genes, and we map this reads to the genes, and we count how many reads and how many expected molecules will be found in our sample. So the thing is that in our case, it's not interested for us, but the genes might have very different length. That means that the longest genes will have more reads that come from them. And in this case, we also need to normalize for the size of a gene. But in the technology which were used in this particular competition, the part of the exact part that which were analyzed was from only the one part of a molecule. So we don't read all the molecule. We read just the small part. So in this case, we don't need to normalize to the size. Still, we need to normalize to the size of how many molecules we took from the cell. I will talk about this a little bit later, but yeah, this is basically what we will see. So in this data, gene expression data, you will meet the table where on rows you have cell barcodes. It's just the names of the cell, we'll say, how they are distinguished inside the sequenator, which molecule came from which particular cell. And in columns, you will have the name of the genes which were covered. I will talk about genes and all the stuff a little bit later. That's okay if you don't understand. If you don't understand the particular map between the names and the, let's say, genomic coordinates. Okay, and now we can move to the CiteSeq data or ADT data or protein data. There was another webinar that I assume you have an access to, but just in brief, we have a technology which can help us to assess the quantity of different proteins on the cell surface. The thing is that we designed and we are producing on a very big scale the special antibodies which can bind the special part of a proteins. So we can be particularly sure, for example, that this, let's say, anti-CD44 antibody can bind to the CD44 molecule on the surface of this particular cell. The thing is that this process can help us to assess the quantity, because for almost each molecule of a protein, I'm sorry, back to our question, back to our antibodies. So let's say we have 5 molecules of CD34 on the surface of the cell, then approximately 5 antibody molecules will bind to them, and that's why then we will make a PCR. We will understand from which particular antibody this molecule come, and we can and we actually can count how many molecules will bind to the cell. So that means that we have a quantity. But you see, there is a problem for gene expression data. When we assess genes, we can read everything. So every single nucleotide chain, we can read it. But with the CiteSeq, we need to design this antibodies. We need to design the PCR handle. The PCR handle is not very important. The antibodies is important. Sometimes you can design very good one and they will work perfectly. Sometimes antibodies does not work very good, and that is just a biological variation behind it. So in the CiteSeq data, as far as I understand, we have almost 130 molecules which were assessed from the surface of the proteins and from the surface of the cells. And I decided to give you a link to the CD to HD HD and C nomenclature. So here you can see the list of all the CD. There was not very good surprise when scientists discovered that sometimes the molecules, the same CD, let's say one molecule, can be different. It can have different glycosylation level. It can have different post-transcriptional modifications. So that is, or it can be different subunits of a different receptor. So that's why this nomenclature is not the best choice, but we already have it, and we made a lot of research on this nomenclature. So that's why we need to deal with this legacy. And I wanted to show you the CD44 molecule, because as far as you remember, this is the main molecule which help us to distinguish hematopoietic stem cells from all the other cells. So you can see here that if this is a location, you will not, but it's very precise, so it's not very useful for you. But here, for example, you can see the other names of this gene and its connection with the other databases. This is important because I hope that that information will give you some hint on how you can select real biological features. So I do understand that all of you will use some kind of regression in your analysis, so in order to make the number of features less. But let's say this particular name, which can be found in the JAX table, can be directly connected with the expression level of CD34. Almost the correlation, I would say the correlation is quite good. So in modern molecular biology techniques, we can hope that the expression level and the amount of RNA actually would correlate with the quantity of protein. Let's say that other things that can be interesting for you also, let's say, yeah, I would say that this is the important part about the names. And maybe the protein resources also might be helpful, because you can find a lot information about the function or other stuff in this thing, where, well, let's say with which biological process is connected this particular molecule, or where it located inside the cell. So that might be helpful for you. Okay, and let's go back to, let's go back to the direct connection between these modalities. So what I want to show you here is that there is, I wanted to show you the connection between the name of the gene. So this is N's, G, and a whole bunch of numbers. This is a special unique ID in in ensemble nomenclature. Here is the different transcripts that you can see from this particular gene. So a lot of genes in human genome produce different transcripts. Most of the time we can select only one transcript, which is most relevant, but that is not, I would say that this is not important for this competition. Also, for this particular table, I selected the coordinates in the genome. This one means that this gene is located on the first chromosome. The second, this one, for example, is located on the 12th chromosome. This is the coordinate of the start of this gene, and this is the coordinate of the end of this gene. And this is the strand. So as far as you know, the DNA is double-stranded, and genes, they can locate it on the different strands. So the first one, or the plus strand, is the same as in the reference genome, and the minus is the opposite one. So why am I and I decided to take this particular example because, you know, in this example we can actually see these three modalities. This is how that will be covered in Site-seq data. This will be connected with the ATAC-seq or DNA data. These particular things should be open. Yeah, and this kind of information could be found in Rnaseq table or JAX table. So this is, and this connection is quite strong. So we can assume that CD34 gene will not be located in other parts of the genome most of the time. Let's say there is some exceptions, but I will not tell about them right now. And also, I put this link here so you can access all this kind of information. I'm not sure if that will work right now because on today's afternoon it experienced some problem. But yeah, let's try to do it like this, but I'm not sure. This is a big database that which covers all types of information between different databases. So I think this is the easiest way to get the different names in different nomenclatures and different for different genes, and let's say, and also you can find here some regulatory features, for example. So you let's say for ATAC-seq data, you might not use just the gene body, but you also can choose the, let's say, other regulatory features. I'm sorry, this is not working properly right now, but you can have, mostly I have an access to this database through the R, but sometimes it's quite easy to do in the web interface. So this link I hope can be very helpful for you. In case it will work, you can find the direct connection between the modalities and features inside your data. Okay, and now I want at first answer questions which were raised. What is a transcript? The first one. You see, the thing is that genes, they are genes have quite complex structure in eukaryotes. They have some exons and introns. Exons are the coding parts, which will actually end up in the mRNA, and introns will not end up in mRNA. I will and transcripts. So, and different transcripts can use different exons. That is not very important in this particular I'm sorry, I'm just looking for quite a quite big picture. Yeah. So this is a picture where you can see different types of splicing, how we can include and exclude exons. So for 4 different transcripts from this gene, we use the cell use different exons. I think that one will not be relevant for this competition, but this is good to know because this is one of the fundamental processes of increasing the diversity of the molecules inside the cell. Vladimir, I'm sorry, I don't know what is not visible. It was quite a few time ago. Maybe I've showed something, but it was not visible for you. Sorry. Yeah. And Sasha just answered the question about the transcript too. Okay.
Maybe your mouse is not visible when if you show something.
No, I don't think. Okay. I think we will be able to answer this question again in case I have not answered it.
But now you are not sharing. Okay.
Yeah,
now
that's because my Zoom doesn't work properly. I'm sorry for this. For now, I think you should see it again. Yes.
Yes. Okay. Thank you.
Okay, so that yeah, and the last thing that I wanted to share with you is the problems with which we experience as a biologist in our own data. So what is the big problem of the biological data for all the machine learning engineers and for us and for us too? The thing is that we have enormous amount of features. This is due to technologies. So we just know how to read all kinds of RNA from the cell. That's why we have all kinds of RNA in our table. And for the typical experiment, we will have around 60,000 features. And it's a very good thing if we have I don't know 2,000 of cells, maybe 10,000 of cells. In this competition, the number of cells is enormous. It's not a typical situation for the science. So we don't have such a big amount of cells. But I do know that machine learners will not like us, especially for this kind of things. Then the observations, we have a very little amount of observations in comparison with the number of features. That is not relevant to this particular competition, but if you will ever work with a biological data, this particular circumstance leads to transposition of tables. So everyone likes long tables instead of wide, and so we have a lot of genes, and we have really little amount of cells. Why we just don't make observations in the columns and features in the rows? So not in this competition, but if you touch other types of biological data, that might be the case. Another problem is that the matrix is very sparse. So we have, I would say we have more than 90% of zeros inside our matrix, and this is not the biological reality. So these zeros are not, that does not mean that this feature is not presented in this particular cell. That means that we just fail to catch this molecule. In a good 10x experiment, I would say that we can catch 10% of mRNA molecules inside the cell. So you can assume that for each particular cell, 90% of the information was just lost. So that's why you might want to make some imputations, let's say, based on other cells of the same population. You can let's say I would suggest that we just take one particular population and we impute the values inside this population. But I'm not a mathematician, not a machine learner, so that might be not a good idea. And the last thing that I want to discuss with you is a batch effect. batch effect is the technical variation that we experience in our experiments. How does it look like when we put all these things together? We have, let's say, one donor and the second donor, and all the cells are so different between each other. So we can assume that there is a big biological variance. But we want to study, let's say, particular population, T cells. And we see in this picture, it's not very precise in terms of mathematics, but in terms of biology, it's quite good. The difference between T cells between these donors is way bigger than the difference between cells inside one donor. And this is not biological true. T cells in all humans actually do the same thing. They have the same function, they have the same molecular background, they have the same morphology, let's say. And all this variation comes from the donors, just because we are very individual and the donors have their own transcriptional background. The bad news is that this batch effect also can be caused by different people who prepare the library for the sequencing, different machines, different days, different, I don't know, the moon phase. So basically that means that when we design the experiment, we should take it into account, and we should collect all the data from the same time and put it in the sequenator at the same time. If we fail to do this, like for example, we have different donors, we can't do this much. We cannot make them the same. In this case, we need to use the batch correction algorithms. I would not say that there is a big consensus in the field which particular algorithm to use, and I actually think that biologists are not very satisfied with the methods which are currently presented in the field. So we are interested in, we as a biologist are interested in studying biological difference. Well, we're actually not very interested in technical variation. So we need this batch effect correction. And that, I assume, will be quite a big challenge in this competition. So maybe the consultation with the previous solvings might give you a clue on this, what to use, and should you use it the same. Okay, so I think I'm ready to answer another batch of questions. Yes, Sasha, so we lose like 90% of each cell. How many cells remaining invisible completely for Rnaseq, and do we care about them? We definitely lose some cells, but we assume that each population will be presented in our experiment in the same proportion. So we assume that we should not care about them. Sometimes, if we are particularly interested in very, very rare population, we use another approaches. So, for example, we are interested in cancer, let's say cancer cells which were left after the chemotherapy. Then, in this case, we will use special enrichment and try to analyze them. But in ordinary situation, we just decide that we catch enough information and we will not care about this. And where is the loss of information more problematic in the Rnaseq data or in Rnaseq data? So I cannot compare, I'm sorry. So as soon as I don't know how much information we actually lose, so where from which we know about the 90%. The thing is that we have not only 10x genomics technology, but other technologies which are more precise, and they catch more information because of a completely different protocol. So that's why we know about this. And I'm not aware of alternative protocols for ataxic data. So unfortunately, I cannot assess this. I'm sorry. And Ksenia, unfortunately, I cannot share my experience from the previous competition because I was, I am now a biologist, and I was a biologist there 1 year ago. So I've just tried to help the ML engineers from our team to understand what kind of data they work with, so they can actually understand. So the base questions was like, what is JAX and how is it's connected with RNA? And you know, my job was to tell this kind of things, because yeah, just because I'm a biologist. But I really need I really need this solving from you guys, because I want to correct the batch effect, for example, very precise, so I can make some biological conclusions on my data. And sorry, I'm not aware of a test train split that was used last year. How we deal with a problem of enormous amount of features. So we actually throw away all the features which have zeros in all kind of cells, and that help us to reduce the size of the number of features for 3 times. So let's say from 60,000 to 20,000 features. And then, even as a biologist, I would like to use some kind of regression, so I can see which features are the best one to describe this. And then I ended up with my biological experience. So I just let's say I'm trying to use the gene set enrichment analysis, because I really understand that if I have some influence on the cells, they will react much. They will not change the expression level of one gene. So this is the biological reality behind it. And I just take the gene set enrichment analysis. I try to connect this with the other types of the response from the cells, and then try to do my biological conclusions. Yeah, but for the ML approaches, I think that the best one was to use some kind of PCA, which was adapted for the highly sparse matrices, and the rich regression shows itself very good. And biologically, the next question is, biologically, it seems that ataxic data from day n might be more predictive of RNA from day n+1 compared to day n, right? I would not say, from a biological point of view, I would not say like this, because I assume that the packaging of a chromosome, it can be very dynamic. So one day, like 24 hours, it's a lot of time for the cell. So I assume that you know, for this type of time course data, that will be very big gap. So I would say that maybe hours between the ataxic data and Rnaseq data will have more predictive power, but in case of days, I'm not sure. I would not try to. That will be not my first clue. Okay, so this is all the questions from the chat.
Larisa, there was a question, I'm not sure you answered. Does the relationship activity was distance to centromere differs for TN5 and polymerase? This from Denis Ostapenko. So have you answered it or no?
Yeah, I'm trying to, I will try to, I hope. Yes, I think that I will not say, yes, I would not say that distance from centromeres will be the predictive for the activity of polymerase, because polymerase starts not from the centromeres but from the origin of replications. Origins of replications, it's known regions of the genome. So I think that we can connect polymerase activity and the origin of replications. What about the transposase activity? I would say that they really may have some correlation between them, because we deal with highly dividing cells, and that means that every cycle they need to duplicate all the DNA, and while the polymerase goes through, it will open some kind of some region of the chromatin. So yes, I think that in this particular experiment, we can experience some correlation between the activity of a polymerase and transposase, especially for highly dividing cells.
Okay, colleagues, any more questions? So let me now try to allow you to unmute yourself so you can ask questions by voice. I hope there are no more Zoom bombers, but if there are any, then we will have small problem, but not big. Okay, so any questions or comments, or you can write in the chat if you want, or you can unmute yourself. And so now there is in the chat from Aurelia, which is the duration of surface proteins on the cell? Is mRNA status analyzed at the same time as surface proteins expected to correlate?
They expect to correlate, but from the last year experience, I would say that loss of information from the Rnaseq data makes this correlation very, very weak. So in general, they should correlate. I would say from 0.7 to 0.9 R squared. But when we took the single cell data, all the dots, they just concentrate around 0, and unfortunately it destroys all the correlation. But biologically we should. That would be very nice to have a imputation, some kind of imputation algorithm which can help to solve this kind of problem.
I'm not sure there should be some very precise correlation because of the post-translational modifications and some other things, even biologically. Yes, so because the
you mean the time of life of particular proteins, so we can yeah, I think the post-translational modification can actually matter in this case. But you know, they will look like outliers, I would say. Then we try to understand. There is some publications about this, the correlation of RNA level and protein level, and in general, it's quite good. So we always know where to find the exception. Let's say erythrocytes. They don't have nucleus, but they can produce new molecules of proteins quite for a long time. Some proteins lives longer and some lives shorter, but in general, the correlation, there is a case of correlation. Yeah, let's say we always need, almost all the time, we need to make the new proteins to, because we do the I would not say we need to renew all the proteins inside the cells. The rate of this renewing of this turnover is very different in different cells. Let's say for the liver cells, it's around 40% of proteins are new every day, let's say. Yeah, but for blood cells, the ratio is another. Yeah, so Aurelia, I'm totally agree with you that even with this with the same epitopes, we expect a correlation.
Yeah, actually, when we see that a cell like shows, say, CD8 on the surface, does it actually usually mean that CD8 transcription level is really high or like was high a day ago, or it could preferably mean that there are some other proteins which push CD8 to demonstrate it on the surface are high? So is it a question of like direct transcription to demonstration like pipeline, or it's much more about some strange regulations inside the cell which decides what a protein to be shown on the surface?
In general, I would say there is a direct correlation with the transcription level, but and it's quite high. And every time when we see the disturbance of some kind of correlation, maybe let's say other transport proteins help to put it into the membrane, we actually consider this as a very we actually need to research very deep this kind of questions. The thing is that they are very difficult to prove. So maybe we just don't know about that this kind of mechanism, this kind of delay, let's say, is very presented, widely presented in the nature. So this is the biological question. I'm sorry, I'm not ready for to answer that, because we don't know how many cases we don't know yet.
Thank you. Yeah, actually, okay, it's okay. I'll try to reformulate more biologically. I mean,
okay, well, I have the question. May I ask?
Yeah, of course.
So your focus of your presentation and the first slides were in bone marrow, and I actually expected that the main goal of this Kaggle competition is not about the specificity, not only about the specificity of bone marrow cells, but rather the prediction of how the cells that differentiate. So what do you think about that?
I agree with you. I would agree with you totally. But you know, I would say that the bone marrow is quite easy to take from a patient, and the cells are very. The cells can resist being in a being without the matrix, so it makes it easier to perform the experiment. I would say that if we take brain cells or other types of cells, it will be way more difficult technically from the first view. Yeah, and differentiation part, yeah, I would agree with you. Also, there is a highly proliferative and very highly divergent cells. I would totally agree with you. That also might be the big rationale behind this competition.
Yeah, so the biological problem that we could see for this data is like how the chromatin closes. while the cell are the differentiation in the process of the differentiation, right?
Yes, that would be, yes, this is the case because different cells need to open and close different parts of the chromatin. Okay, Sasha, I think that we need to put it a little bit more straight. When we measure the protein level with Cytosec, that does not mean that we measure the epitopes which are presented by HLA. The thing is that we just measure the surface proteins. Let's say CD44 is the adhesion molecule which helps cell to be connected with other cells. In this CD nomenclature, we can see, I believe.
Yeah,
Yeah,
thank you. Yeah, actually, you answered the question. Thank you. Yeah, it was I have very random knowledge in biology. Sometimes they coincide with reality, sometimes not. I understood only now that there are two kind of two words about a cell present something, and one of them is about the immune activity, and other is about the stuff when the receptors are go. And I do not know, can they interact? I mean, if we have good antibody for CD8, can it also detect the CD8 presented in HLA?
I think that depends on how the antibodies were formed, because if this antibody can recognize the part where the epitope is located, then it might disturb the connection. But most of the time, it will the antibodies will be connected to the other parts of the molecule, so we can actually cope, but they will not be, this interaction will not be disturbed.
Yeah, but we don't know what proteasome will decide to give to HLA to present. It's kind of random process. So actually any part of the receptor could be presented also. It's just like I do not know.
Yeah, but the different antigens are presented on just only on HLA molecules, but we measure other types of molecules. They are not in almost all the case it will be adhesion molecules, by the way, or some receptors, and they just they are not connected with the process of antigen presenting. I actually don't know how the HLA will be named in CD nomenclature. I need to check it.
Thank you. Yeah, thank you so much.
And what is HLA, which you asked the question? This
is a human leukocyte antigens, and this is the protein complex which not only is not only exposed on the surface of the cell, but it's also take with this the parts of the antigens. And on this HLA molecules, the immune cells show each other the antigens which we need to attack. There might be some virus proteins, actually peptides from viruses. There might be something from the bacteria, and let's say some T cells, just some dendritic cell. It's actually dendritic cell. It shows it on its surface and say this you need to attack, and T cells and B cells, they say okay, and then they make all this type of immune reaction to build the whole defense system against this particular pathogen.
Okay, thank you. Any other questions or comments, suggestions, points to discuss, or anything?
I have a question, or I had a question. Can you show that table where we match attack-seq and Cytosec data?
This one?
Yes, this one. I'm pretty sure that you draw something to show how to match this data, but it's not visible for us, or at least for me.
It's this green. Can you see this green line?
Yes, I can.
Yeah, this is the name of the gene, and that's how it will be named in Rnaseq Data. This is the coordinates on the chromosomes.
So if you are pointing to somewhere, I don't see it.
No, I'm drawing with the annotate function and do it with this green line.
Perhaps because annotation is disabled, we can see it, or at least I can see it.
Okay, just maybe
Yes, we now see something. Yeah, we see the arrow. You can just show, for example.
Okay, this this red thing shows the RNA level, and this is the basically this is the name of the genes, and they will be named the same in the table of JAX. Maybe in a different nomenclature, but in Biomart we can do it. Okay, so the red one is RNA. The next one, I'm doing this now just in a presentation, so I hope that you are able to see it.
But Vladimir, I think it's not about attack-seq data. This is about the site-seq. It's not.
Yeah, this is actually this is the question: how to connect this. So
this is the most important question, I think.
Yeah. So the red one.
Is there anything related to attack-seq data here on this table?
Yes, there will be this blue line. This blue shows the particular coordinates on a chromosome. This is the chromosome number, the first one. This is the start.
Gene. For the
yes.
gene, yes.
Yes,
okay,
and gene should be opened in order to allow the expression. So this region should be open if we want to see the RNA level and if we want to see the proteins. So this is the connection.
I would say this is maybe necessary condition, but not sufficient, because we need to open some promoters, enhancers, and so on, which can be quite far from the gene.
Yeah, that's true. That's why I hope that the data which is published in Ensemble regulatory features and some other resources will help you to understand that this gene does not only require this chromatin to be open, but also the regulatory features. But the regulatory features should be downloaded and should be researched separately.
Okay, thank you.
And how do we understand the strands? Oh, okay. As I've got it, strands are different sides of DNA.
Yeah.
But how do we work with it? Are there some differences between positive and negative strands for the same, for example, CD34?
For CD44, no, because it's located on a negative strand, and that means that we will the polymerase will read it from one side to another. But in terms of the data, that will not make any difference, because we can understand, we can assess only the open state of a chromatin in Rnaseq data. And we can't just open one side of DNA. We need to open both strands. So in this case, no, we don't need the strand information. But the strand information help us to understand in which direction the transcription will go. So the transcription is always from one end to another end, and the strands are anti-parallel. So that means that if gene is located on plus strand, the transcription will go, let's say, from right to left one, now to left end. And if the gene is located on the other strand, that means that promoter will be, in terms of the coordinates, the promoter will be after the gene. Am I sounds understandable?
Looks like that. So if for positive strands, for example, we go from lower coordinates to higher, and vice versa for negative.
Yeah.
Oh, yeah. Thanks.
So basically, for this particular gene, this is the start of the gene, but end of the coordinates. Everything is flipped for the negative strands. Okay, guys, no more questions in the chat. Any questions by voice?
Larisa, thank you very much for the presentation. So let me ask a question. So, but this strand information, is it related to attack-seq data? Or, I mean,
No.
for example, can you predict the strand from the attack-seq data? Or,
No, because the whole double helix will go onto the nucleosome. So if we open it, we open both strands. But promoter will be promoter for the genes which are located on a plus strand. They will be before the genes in terms of our coordinates and in terms of attack-seq data. But promoters for the genes on a minus strand, they will be after. So that is important only for the regulatory features, not for the gene bodies.
Okay, thank you very much. Colleagues, any other questions or comments or suggestions or whatever? Okay, so thank you very much. It was a great presentation. Thank you for sharing your experience.
Thank you, guys. Very fruitful discussion. I was very glad to present it, and I'm always open to answer the questions in Sberloga chats and in direct messages also.